In this video, I will be discussing about the first component of the cytoskeleton, the microtubules. So microtubules are actually rod-like in shape, but they are actually hollow. So as we can see in this illustration, their components are actually to the sides, and in the middle, it's actually hollow. So here's a picture, an electron micrograph of microtubules. So these are actually hollow rods. So how many rods do we have here? Each one of these are actually known as protofilaments. Okay, so if we count, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 protofilaments per microtubule. Alright, so you can see it here in the electron micrograph. They are very fine and they run along the length of the microtubule. So here's a more detailed illustration of the microtubule showing its dimer monomers. All right. So the dimer monomers are made up of these alpha and then beta tubulins. So we can also see here how the whole microtubule is folded so that it has that circular shape. Okay, and you can see here in the middle it's hollow. So there is your dimer. It's actually made up of beta and then an alpha tubulin. All right. Now, there is a portion here which is containing GTP, and only the one found in the beta tubulin is exposed so that it can react for polymerization. And so the addition of new components of the microtubule, if ever it needs to become longer, is only possible on this plus end where the beta tubulin is present. So notice here in this illustration, we have alpha, beta alternately. And here, all of these ends here are all beta tubulins. So this is the alpha, the positive end of the microtubule, and this is where the microtubule will still continue growing. Okay, so remember the monomer of microtubules would be combined alpha and beta tubulin. So we call these dimer monomers. And take note, the way of folding is actually not level. You can see here there is a seam happening between these two ends of the folding. So each one of these beta alpha, each one of these lines will be the protofilament, and there are 13 of them in one microtubule. And so because of this unleveled folding, okay, the folding is not level between these. This is actually an asymmetric shape. Asymmetric also in a sense that one end ends with alpha and the other end ends with beta. So therefore, it's an asymmetric structure altogether. So microtubules are asymmetric. There's a negative end and a positive end. So that is very important in terms of how the microtubule works with motor proteins. And then we also have the microtubule associated proteins, which of course understandably associate themselves with the components along the length of the microtubule, as we see here. So microtubules function mostly in structural support and organization. Okay, so as we see here in this example, this is a circular cell, okay, that has a circular shape. The orientation of the microtubule will be from the center near the nucleus going outwards towards the periphery of the cell. Okay, so this is for round and flattened cells. In columnar epithelial cells, the orientation is that from the top of the cell going downwards towards the nucleus of the cell. Okay, so that's the orientation of the microtubule in columnar cells. In cells with projections like neurons, the orientation of microtubule is from the cell body along the axon or the projection going away from the cell body. In plant cells, you remember this illustration? Okay, you remember why the microfibril made by the cellulose synthase follows a definite pattern. Okay, they're all parallel, and that's because the microtubule underlying the arrangement of these cellulose synthase actually dictates also for the movement of the microfibril that is being secreted by the cellulose synthase. So the microfibril orientation follows the orientation of the microtubule underneath the plasma membrane of the plant cell. So here's a picture of the color-coded microtubules in a plant cell. You notice how they are arranged so that they are perpendicular to the overall length of the cell. So say, for example, the cell is long from the bottom here to the top portion here. The microtubules are arranged perpendicular to that, cross-sectional. And that's important because as the plant cell grows, as the central vacuole fills with cell sap, the plant cell can actually grow, can elongate in a certain direction only. As you notice here, the expansion is only in this direction, and that's thanks to the orientation of the microtubule that has influenced the orientation of the cellulose microfibrils on the plant cell. So it's more difficult to expand, say, in this example, from left to right. So the expansion is really top-bottom expansion. So along the grain. So another function of microtubules is also for the organization of the organelles. So here we have an example, a cell. And notice the stacks of the Golgi apparatus here are anchored to these two microtubules. So what happens if the microtubules will be absent? If there are no microtubules, or if the microtubules are not organized at all, the stacks of the Golgi apparatus will also be disorganized. So you see, you need an organized microtubule, the cytoskeleton, for an organized organelle. In this case, the Golgi apparatus. Then another major role of microtubules, because they have a negative and positive end, is in intracellular motility. And that's the movement of vesicles and other organelles within the cell. So, as I mentioned before, they serve as railways for the moving vesicles and organelles, which themselves are moved by motor proteins attached to them. So here we can see vesicles that are carrying neurotransmitters in a neuron. So from the cell body, it goes into this end where another neuron is waiting. But there is also an opposite movement where it can move from this portion towards the cell body. So what are the motor proteins involved? If you can remember, the first one would be kinesin. So they require ATP in order for them to move, and they are unidirectional in movement. So kinesins can only move in one direction, and this is their structure. So there is a portion here, the foot, okay, and then there's the portion here where they actually carry the vesicle. So the vesicle is attached here, and then this is the foot that walks on the microtubule. Or rather, it's called here the head. Alright, and the tail is the one that holds the vesicle or the organelle. So very small, they are around 80 nanometers from head to tail. So this is how they move. Okay, there is sort of a catwalk-like movement between these different heads. Okay, so as you can see here from the top view, the kinesin is moving along the microtubule as if it's a supermodel walking along the catwalk. And the direction of movement is from negative to positive, and that alone. So it cannot move backwards. It's unidirectional. So in the neuron, for example, the movement from the cell body to the end of the axon is in a negative to positive direction. That's the direction of the microtubules. So it will be the kinesins that will carry the vesicles that contain the neurotransmitter to move along in this direction. And then there are other kinesins. There are actually 14 of them. Okay, but not all of them have exactly the same function. For example, kinesin number 13 is not capable of movement, but it acts as an enzyme to depolymerize microtubules. So microtubules can either be assembled or disassembled, and the disassembly is done by kinesin 13. So the question now is this: if kinesin can only move from the negative end to the positive end of the microtubule, what accomplishes the work of the movement of vesicles from the positive to the negative end? The answer will be another motor protein associated with microtubules known as the dynein. So the cytoplasmic dynein in particular. So they are found in cilia and flagella. They are more conserved than kinesins, meaning to say there are very few types of dynein, particularly just cytoplasmic and those that are attached to the cilia and flagella. So around 2 at least. And then they move from positive end to the negative end of the microtubule. So this is the answer, the opposite movement for the vesicles. And they look like this. They are much bigger motor proteins than the kinesins. So you have here the head, but then there is a stalk, so it's sort of like the kinesin but with a high heel. Okay, so there's a high heel, so it can do proper walking on the microtubule from positive to negative end. And this is where the vesicles are attached. So here we have a vesicle that's moving from the negative to the positive end of the microtubule, and the ones that are carrying it, of course, this time are the kinesins. But attached to the membrane of that vesicle is also a dynein, but here it's quite inactive because the movement here is from negative to positive. But if ever this vesicle needs to move in an opposite direction, it will be the dynein now that will be active and the kinesins will be inactive. So now you should be able to understand how the cytoskeleton, particularly the microtubule, serves as a railway for the movements of organelles and vesicles in different directions inside the cell cytoplasm.